Our next speaker is Maggie Diguardo from the Mayo Clinic. Maggie is our second place student trainee award winner, and the award has been graciously provided by Seattle Cancer Care Alliance. Maggie's title is RNAseq Analysis of PD-L1 Expression in Colorectal Cancer and Endometrial Tumors in Correlation with Microsatellite Instability.
Hey, good afternoon. So I'm Maggie Diguardo, and I am going to talk to you today about utilizing RNAseq. And actually, I'm going to follow up a little bit on what Scott just spoke about in terms of immunohistochemistry and possibly a different method of measuring PD-L1. So we looked at this in colorectal and endometrial tumors, and then we correlated it with microsatellite instability. So we're going to look at a different subset of tumors in this talk, but a lot of it will kind of correlate with what Scott was saying. And my apologies for some of the redundancies. This one? Okay, thanks. Sorry, so I'm a little handicapped. Okay, so I have no disclosures. Except that I'm using my left hand for everything today. So my objectives are to briefly give a background on PD-L1. My apologies for the experts in the room, but just to equilibrate the audience, we're going to talk about the mechanism briefly. I'm going to discuss PD-L1 as a biomarker, and I'm going to correlate this relationship to mismatch repair and microsatellite instability. I'm sure you guys are all aware of the paper that I'm going to reference here. I'm going to talk about how we designed our project, the results that we have thus far, and then our future goals. So we're going to start quickly with PD-L1. We all know it's a transmembrane protein. It's encoded by the gene CD274. This is going to be it's expressed on antigen-presenting cells typically, and it's a method for evasion, so it inactivates the T cells. So in the setting of cancer, this is a very valuable mechanism in that as the tumor cells are mutating, they're generating new epitopes. These epitopes are going to be presented by the MHC to the T cell receptor, and then that T cell, if it deems it foreign, will activate and theoretically should attack the tumor cell. So essentially, however, if this tumor cell also expresses PD-L1, and this binds to its receptor PD-1 on the T cell will inactivate, and now you have immune energy. The checkpoint inhibitors that were discussed earlier are monoclonal antibodies that target either PD-L1 or PD-1, as we know, and if you block this interaction, you reinvigorate the T cell. It can target the tumor cell and do what it was supposed to do and kill it. So it would seem intuitive that we would measure PD-L1 as a method of including these patients in these for these targeted therapies. And we do. We measure it by immunohistochemistry. And as it's been noted, there are multiple benefits to this. However, with immunohistochemistry comes multiple limitations. And these limitations stem primarily from the fact that as these targeted therapies have gone through clinical trials, so have their companion diagnostics. And although some of them don't have a companion diagnostic, many of them do. So these antibodies are different for each one of these tests. And as we see, this leads to no standardization in the scoring metrics. And this includes both the cutoffs that are used and the cell compartment that's targeted. So I think it's important to note that primarily these assays are looking at the tumor cell compartment. However, some of them are incorporating the immune cell. If you look at the clinical trial for atezolizumab, you'll see that what they found was that positive scoring in the immune cell was actually a better predictor of response than on the tumor cell itself. And so they have incorporated immune cells into their scoring metrics. This is important in colorectal tumors because, as I'm sure you all know, they often demonstrate a large immune cell infiltrate, and typically they show zero staining on the tumor cell. So as with any IHC, you're also relegated to variation in interpretation and tumor heterogeneity. So this is a topic of much debate, how immunohistochemistry should still play a role, and there have been multiple studies that have been targeting this very question. So the one I bring your attention to is this Blueprint PD-L1 IHC assay comparison project. It was a collaboration of multiple groups, and it was led by a group out of University of Colorado. There are 2 phases. I'm presenting only the data from the first one. But essentially, what they did was they looked at 4 of these assays, and they had, I think, 38 patients, or 38 cases, 4 assays, and they had pathologists score them, separate pathologists scoring both the immune cell compartment and the tumor cell compartment. And what they found was that looking at the tumor cells, 3 of the 4 assays were closely aligned, but 1 showed significant variation. In addition, when they looked at the immune cells, they showed variation across all 4 assays. And so what this means for our patients is that interchanging these assays and cutoffs leads to misclassification. And that's a big deal for these patients. And it goes both ways. Either you're giving them immunotherapy that they're not going to respond to, or they're missing out on a checkpoint inhibitor that could be helpful. So this begs the question, actually it begs multiple questions, but one of which is there a better biomarker? And so I'm sure you're all familiar with this paper, which came out in 2015. This group, Diaz's group, was able to show that in colorectal cancers, microsatellite instability was actually a better predictor than PD-L1 of response to these anti-PD-1 drugs. And so I guess everybody's kind of, and this was, I'm sorry, but these findings were so significant that it led to the first tumor-agnostic drug approval in May of 2017. So now in colorectal tumors, what is the relationship between microsatellite instability and PD-L1? So we had the same question, and in looking at microsatellite instability, obviously it's a surrogate marker for mismatch repair deficiencies, and we see those primarily in colorectal and endometrial tumors, although that list is not exhaustive, and we all know that it includes, we can see microsatellite instability in multiple tumor types. So we started with 36 endometrial and colorectal because they're the primary tumors. We had FFPE for all of our cases, and we had a paired normal for each one. All of these went through pathologist review, and so we did ancillary testing as applicable, and that would include microsatellite instability testing and hypermethylation studies. Then we did RNA extraction, we followed through with our QC, and then we included and excluded our cases based on their DV200 scores. So the DV200 is going to be the amount of reads that are greater than 200 nucleotides. It was part of the algorithm put together for the platform. So essentially, after this, we ran them on the TrueSeq RNA axis if they fulfilled all of our criteria. And then we quantified the differences in mRNA expression, and we did this as a fold change between the tumor and their normal counterparts. 18 of these 36 cases had enough tissue for us to complete the PD-L1 IHC. We used the antibody E13LN, and these were reviewed by 2 separate pathologists. So all of these samples were de-identified. There was no access to the RNAseq data when the pathologists reviewed them. And then we arbitrarily, because there were no real good cutoffs, and essentially which cutoff do you use for colorectal, we categorized them by 0, less than 5%, 5%, and greater than 5%, and then we scored them. So my sample distribution was such that, again, we started with 36 endometrial and colorectal. We separated them by microsatellite categories, status. So we had 10 or 11 that were microsatellite stable. These we used as our controls. And we had 25 that were microsatellite high. Of the microsatellite high, as you guys probably know, you can get to this pathway in 2 ways. We had our hypermethylated group, which was our somatic alterations, and we had 7 representatives of this group. And then in our germline, we had 18 cases, and we were able to show cases from all 4 of the primary genes that are involved in the germline changes. And the numbers for those are represented below. So here are some of our results, and essentially what I'm going to do is bring your attention to first the y-axis. You can see it's the CD274 fold change, and then the groups are color-coded. So the first group I'll draw your attention to is going to be the MLH1 germline, and those are the brown bars. And what you can see is that anything above 0, again, means that there was an increased expression in the tumor when compared to its normal counterpart. And as you can see with the germline cases, for MLH1 at least, those are primarily showing an increased expression in the tumor. I'm going to skip the pink group for now, and we're going to move to yellow, which is MSH2. These guys were only 2 cases, but both of them showed increased expression. MSH6 was similar with 1 outlier. PMS2, interestingly, all of these showed increased expression in the tumor when compared to the normal, and these showed the highest amount of expression out of all of our cases. Highlighted in black are our controls, so this is the microsatellite stable group, and you can see they're all hovering right around the 0 line, less than a 1 to 2-fold change. And these show a very similar pattern back to our pink group, which is the MLH1 hypermethylated group. So these were some interesting findings. So looking at these from a statistical perspective, we did simple t-tests, and instead of doing them by groups, we categorized them by our germline in total, and we compared germline to our microsatellite stable cases. So when we did that, what we were able to show was that the germline group of tumors, microsatellite high, show a statistically significant difference in CD274 expression when compared to the microsatellite stable cases. And that we expected. That makes sense. It seems like an intuitive step. But what was interesting was when we looked at our germline group and we compared those to the microsatellite high hypermethylated cases, we also showed a statistically significant difference in CD274 expression. You can see, too, that the hypermethylated group and the microsatellite stable group show no difference, no statistically significant difference in expression. So this was really interesting because if microsatellite instability is a better predictor, one would expect that you would show higher levels of PD-L1. Both of these groups are microsatellite high or unstable at high levels, 5 out of 5 markers, and yet you're showing very different levels of expression. And you can see down below in the right-hand corner, the means are listed right there. So the hypermethylated group was at negative 0.08, and the average for the germline group was 2.43. So for my last slide, I'll just show you how this kind of plays out when looking at the immunohistochemistry. And this is, you can see in the first column, I've got the PD-L1 immunohistochemistry on the tumor cells. The second column is the immune cell staining scores. And the third column is the CD274 fold change that we see. Again, you can see the distribution of colorectal and endometrial, and there was really no differences between the tumor type. But as you can see, and I'll draw your attention first to this category. So if you look at the CD274 fold change column, you can see it's in increasing order. And what we see is that actually the IHC is trending to follow the same pattern. So no staining on the tumor cells yet, and on the immune cells you see primarily less than 5%, with the exception of 1 outlier in my hypermethylated group. And you can see that it doesn't really, these are mostly our negative cases showing increased expression in the normal, and they reach about a 1-fold change. Right around 1, if you come to this group, I'll draw your attention to, all of these cases with the exception of 1 case in this cohort show 5 or greater than 5%. So you can see that the CD274 fold change is just increasing with this. And then the last group I'll bring your attention to is this one down here. And you can see this is where the tumor cells start to stain, and this is where we have our highest levels of CD274 expression, going from a jump of about 2.6 to 3.6 all the way up to 9.56. But importantly, I bring your attention to our outlier in this group, and this is really important because these are one of the cases that Scott was mentioning earlier. So if you see this third, I'm sorry, I lost my arrow, but if you look at the second to last case, the PMS2, what you can see is that there's 0% staining on these tumor cells. So if you were to use just the tumor cell staining, this would be negative, but it's showing a 7-fold increase between tumor and normal of CD274 expression. You have other cases you can see in my hypermethylated group. These are microsatellite high, but they have very low levels of CD274 expression. So would these be one of your patients that's getting immunotherapy, you're exposing them to multiple risks, and they're not going to respond? So while I can't draw any statistically significant conclusions from this data yet, due to our small numbers, I do see a trend, and I think it's one that will continue as we continue to add cases to this group. So what we can say that we learned, or that we have learned thus far, is that the MSI high tumors with germline defects show an increased CD274 level of expression when compared to microsatellite stable tumors. Again, that's what we expected. What we can also say from what we've learned thus far is that the microsatellite high tumors with germline defects show increased CD274 expression when compared to hypermethylated tumors that are also microsatellite high. We can also say from this data, at least in our cohort, that the hypermethylated cases and the microsatellite stable cases show no significant difference between the two groups. So where are we going to go from here? What are our next steps? Well, we're just increasing our numbers. We have a target of 60, and we'll be getting data back from those shortly. We would like to include more tumor types because I think it would be interesting to see how this plays out with other tumor types that are microsatellite, that show microsatellite instability. And then eventually, because it's a biomarker, none of this really matters unless you can correlate it with outcomes or response to these therapies. And so if you're going to be able to use RNAseq to if you're going to be able to validate RNAseq as a probable method to measure PD-L1, we'd have to do it with, obviously, therapy outcomes to establish cutoffs. And so that's all I have. We have some more work to do, but it's pretty interesting, and thank you for your attention. And thanks for all of these guys. Sorry. Most importantly.